Hello, everyone. Welcome to Being Patient's live talk. And today we'll be speaking about the role of our brain's immune cells called microglia and how it could be involved in Alzheimer's disease and open up new ways to effectively treat Alzheimer's. And today we have Jason Ulrich, Associate Professor of Neurology at Washington University in St. Louis. So Jason, thanks for joining us to talk about your research.
No, thanks for having me.
Yeah, so start off with Jason, can you tell us about what is the microglia and why is it important for our body and our brain function?
Right. So microglia are what is called a resident immune cell in the brain. So it's basically an immune cell as part of the innate immune system, which is part of the immune system that isn't, antibodies and antigen specific, but it's just a general defense mechanism. And there's been a lot of identification of genetic risk factors for late onset Alzheimer's disease in the past 10 years or so. And one common feature of a lot of those genetic risk factors is that they're genes that are found specifically expressed within microglia in the brain. And so this is sort of pointing to a role for this cell type in the brain as somehow affecting the disease process.
Mm-hmm. Right. So the microglia, they're a type of immune cells, right? And the way I've read it is that they clear off like toxins in our brain, right? In a way, is that correct?
Yeah. So they do probably a lot of different things. They probably help maintain myelination within the brain. They defend against infections as any innate immune cell would. And in terms of Alzheimer's disease, it's still not entirely clear what purpose they serve when and what is beneficial and what is harmful.
Right. And you mentioned myelination. What does that mean?
Oh, myelin. So your brain has, of course, white matter and gray matter. And the white matter is heavily what's called myelinated. So it's a thick membrane wrapping around the axons that helps neurons communicate with each other.
Right.
And so this requires a lot of maintenance and regulation, and microglia may be involved in that.
Right. And the microglia, it's part of this family of cells in our brain called the glia cells, right? Can you tell us about, like, in a broader term, like what are glia cells and like how microglia fits into this?
Right. So if you think of the brain, if you want to cut it into two parts, you have your neurons, which fire action potentials and signal to each other. And then you have a host of other cell types in the brain that are also extremely important. And those are called glia. And you can break down glia into different subtypes. There are cells called astrocytes, which do any number of functions of supporting neurons through providing energy sources or clearing out transmitters. You have cells like oligodendrocytes, which form that white matter myelination around axons. And you have microglia, which again provides sort of that innate immune defense and can also be involved in sort of even shaping how neurons communicate with each other through clearing out synapses that aren't used and things like that.
Right. And as you and your colleague wrote in the Scientific American, right? Like back in the day when Dr. Alzheimer was looking at brain autopsy and found clues of Alzheimer's. There were some clues about like abnormalities in the brain's glia cells. Can you tell us a little bit more about that?
Yeah. So buried in that initial report that Dr. Alzheimer made 100 plus years ago, he noted what we now know as amyloid plaques, which are those deposits of the peptide called A beta that form in the brain very early on in the development of the disease. And he noted inclusions of what we now know as a protein called tau, which form these fibrils inside neurons. And that's really correlated with the advanced stages of the disease when you start getting sort of memory problems. And one thing that he noted that kind of took a back seat for a number of decades was that there were changes in the glial cells, what we now know are microglia or astrocytes, sort of around that amyloid plaque that he found. And there were increases in like fats within these cells and just sort of changes in their structure. They didn't really have stains back then like we do now with antibody stains and everything else to identify specific proteins. But he knows that they were larger and that there were structural changes in them.
Right. And do you have any thoughts about like why, his observations of the glia didn't receive as much like attention as the plaques and the tangles?
Well, I think, to be fair, Alzheimer's is a big problem. And if you're going to start working on it, obviously working on those two proteins that start aggregating is a very, it's a very logical place to start. And understanding how A beta plaques form and how tau tangles form and how those relate to each other and how these proteins are made and degraded and everything is very important. And again, until the identification of some of these genetic risk factors that were expressed within microglia, right, it wasn't entirely clear if the microglial response was simply, secondary to the development of the pathologies with the A beta and the tau, or whether they were actually participating somehow in that. So,
right. And so what have been some of the genetic risk factors that have been shown to be related to microglia and Alzheimer's?
Right. So there's been several. Literally the two, the most common or the most important one that really transformed, I think, the field of microglial research in AD was a protein called TREM2, or a gene called TREM2. And this is expressed within microglia only in the brain. It's also expressed in different peripheral innate immune cells that serve similar functions in their respective organs. But within the brain, some researchers identified these mutations in this gene that decreased the gene's function. Okay, and the gene had previously been implicated in another disease, very rare disease called Nasu-Hakola disease, which is a very early onset form of dementia and has bone loss and everything else. But the variants that were identified in Alzheimer's were different than these. So Nasu-Hakola was just a complete loss of function knockout. And these, if you only had one copy of a slighter loss of function mutation, it increased your risk of developing Alzheimer's disease about 3 to 4-fold, which was pretty significant. So our lab and several other labs, Bruce Lamb, Marco Colonna, probably leaving some people, Christian Haas, a lot of people started working on this and trying to understand how losing TREM2 function within microglia could influence this disease. And so one of the first experiments that was done was to take our mouse models of Alzheimer's disease, okay? Now there's no true mouse model of Alzheimer's disease because there's not a mouse that gets amyloid, tau, and neurodegeneration, but you can model different aspects of the disease in mice. So one of the tools we have are mice that develop amyloid plaques. They've been engineered using human proteins to develop these plaques. And when we crossed these to mice that don't express TREM2, we found that normally you get microglial clustering around the amyloid plaques. And it's not entirely clear what they're doing at that point, but you see a lot of microglia around the plaques, and they're definitely responding to it. And this is seen in human AD as well. Without TREM2, that clustering was totally gone. It was like the microglia weren't seeing the amyloid at all. And we observed this, and many other labs around the world have observed this. And this seems to be one of the major phenotypes of this loss of function.
Right, got it. And how about with what about with tau pathology? We talked a little bit about amyloid pathology. What's the relationship between TREM2, microglia, and tau then?
Yeah, so now it gets extremely complicated. So if you say take a mouse model of tau pathology, what we have found in our studies is if you have a mouse model that develops sort of tau tangles and neurodegeneration, if you cross those to a mouse that doesn't have TREM2, you actually see reduced neurodegeneration. And this is going along with a reduced change in the microglia. So generally within our tau mouse model, when the microglia are in the presence of this tau pathology and this neurodegeneration, they become very activated, for lack of a better word, and they express a lot of genes that were responding to the damage in the plaques. And this is really driving a lot of the neurodegeneration in this mouse model. Now, when you knock out TREM2, you don't see that change in gene expression, and it's actually protective in the tau mouse, okay? But if you take a mouse that has amyloid and you say inject tau isolated from human AD brain, one facet of tau that we've come to understand over the past 15 years or so is that it can template its own misfolding. So you can take sort of a misfolded species of tau, and it'll corrupt other tau that is normally folded perfectly fine and doing its job in the brain. So if you inject one of these bad seeds into a mouse with amyloid, but no TREM2, it dramatically increases the amount of tau pathology around amyloid plaques, suggesting that part of what TREM2 is doing is to protect the brain around the plaque from sort of allowing this misfolded tau to accumulate in Alzheimer's disease. And another group led by David Hansen published that if you had a mouse that was genetically engineered to express both amyloid and tau pathology, again, if you knocked out TREM2, the tau pathology, it became far greater, far earlier than if you had TREM2. So there seems to be something going on with how TREM2 is sort of mediating that relationship between amyloid and tau pathology.
Got it. So there's a very complicated, seems to be a very complicated relationship between TREM2.
There really is. And there was a paper that came out just a few weeks ago, which complicates it. This is all in mice. And you see some of these facets in humans. You can take humans that have these, these TREM2 variants that have Alzheimer's disease. And you can see some of these facets of like more tau pathology around plaques that we see in mice. But it's very difficult to fully take what we see in mice and try to investigate it in humans because everything's end stage or correlational. There was an interesting paper that came out a few weeks ago suggesting that microglial activation is also correlated with the spread of tau pathology throughout the brain. So in that case, correlation is not causation, so it's difficult to interpret, but it's always important to keep in mind trying to take your observations from mice into humans. And I think there's a lot more yet to learn.
Right. So there's still a big gap between what we're finding, what you're finding and scientists are finding in the mice studies, and then what we can infer.
Yeah, I think bridging that gap is a big question.
And what about like with stem cell research and with like iPSC models? Are there any type of research that's yielding findings in this area then? Yeah,
so there's a lot of people that are working with what's called inducible pluripotent stem cells, where you take basically skin cells from individuals with whatever variant you're investigating, and you can engineer those to turn into different cell types. So you can turn those skin cells, you can de-differentiate them and then turn them into neurons or astrocytes or more recently microglia. And there's a scientist in California, Matthew Blurton-Jones, who's developed a tremendous model where he takes these inducible microglia and transplants them into mouse models. And so he can sort of study a human microglia-like cell within the context of a mouse. And reassuringly for our mouse studies, what he sees really recapitulates to a large degree what we see with sort of TREM2 knockout or TREM2 loss of function of the mouse gene. So there are a lot of similarities, and human microglia may have a few distinctions that we're still trying to understand in terms of the exact gene expression profile they turn on as it compares to what we see in mice.
Right.
But there's a lot of good work being done in that area.
And maybe further complicating the picture, I wanted to ask you about is also the APOE gene. So have what have been some of the studies in terms of looking at the relation between like APOE and microglia and Alzheimer's?
Right. So APOE is it's the apolipoprotein E gene. So this is a gene that it's a lipid transport gene. Now, normally in the brain, it is the predominant lipotransport protein in the brain, and it's largely expressed by astrocytes normally, which are one of those glial cells. And it probably does a lot of different things. It shuttles lipids from astrocytes to neurons and sort of maintains all of that lipid homeostasis, the healthy lipid levels and everything in the brain. Now, when you start to get a neuronal insult, a neural degeneration, something like that, microglia can strongly upregulate their APOE expression, almost to a level equivalent to astrocytes, at least in our mouse models. And this seems to be important for a very similar gene expression profile that TREM2 is regulating. And so these seem to be maybe not signaling to each other, may or may not be signaling to each other, but they seem to be within a common program that these microglia have installed to respond to neuronal damage. So for the long time now, and I should mention, of course, APOE is probably the strongest, most robust genetic risk factor for late onset Alzheimer's disease. So if you have one copy of APOE4, you're increasing your risk of AD by, I think, about 4-fold. And if you have 2 copies, it's about 12-fold. And it does this in a number of ways that we're still trying to understand. We know that individuals with APOE4 develop amyloid plaques sooner and more abundantly than individuals with other copies of APOE alleles, which are 2 and 3. And APOE3 is probably the most common of those. Interestingly, APOE2 allele is actually protective. So if you have an APOE2 allele, it protects you from developing Alzheimer's disease. Now, we see this in mice, we see this in humans, that it affects amyloid pathology. More recently, our lab has identified that APOE can also influence neurodegeneration in response to tau pathology. So if we take mice that have tau pathology, develop neurodegeneration, and we cross those to mice that express one of these human APOE proteins, right? 2, 3, or 4. The mice that express APOE4, this risk allele, develop more neurodegeneration, more severe tau pathology than those that express other APOEs or don't express APOE at all. So you can also see sort of a more rapid decline if you look in individuals and look at how fast their brain regions with tau pathology are sort of degenerating. They seem to degenerate more quickly in people that have APOE4. So part of this may be going through the immune system, we think. Although it's again not entirely clear which APOE, which cell type APOE is coming from that's mediating this effect, whether it's APOE derived from astrocytes, derived from microglia, doesn't matter. And these are all open areas of investigation right now.
Got it. And how about for your lab then? What do you think are like the most, like if you had to boil it down, what do you think are the most important questions that you have to answer in order to really develop like effective therapies that can target microglia and really help patients?
Right. So probably the number one issue with developing a therapy that would target microglia is what would you want to do to the microglia at which stage of the disease? So if we just take our preclinical mouse data at face value, it would suggest that with amyloid, right, if we don't have functional TREM2, you get more damage. So maybe if an individual has amyloid plaques but hasn't really progressed to a lot of tau pathology in their brain, you'd want to activate TREM2. You'd want to sort of drive that protective microgliosis around the plaque. Now, if an individual had started to develop tau pathology, would that still be effective, or would that actually make things worse? That's kind of an open question that we don't really understand yet. But you know, I think microglia are a tricky cell to target, and there are some folks working on some clinical trials in humans using, say, antibodies that turn on TREM2
Right.
to try and figure out what's going on.
Right, and your lab is also currently developing some antibodies targeting microglia.
Yeah, so we've developed antibodies, and I'm a I forget the word, co-inventor on a patent for TREM2 agonist antibody, along with my PI, David Holtzman, and staff scientist in the lab. And our goal is to see if, you know, turning on TREM2 and amyloid models, or tau models, what effect that has on neurodegeneration or amyloid pathology and microgliosis.
Right, so it'll probably be quite some time still before it can enter human clinical trials, you think?
Oh yeah, yeah. There are, like I said, there are antibodies in human clinical trials right now. At least one I think is doing clinical trials, but yeah.
Right, and as we talked a little bit about this before this interview, you mentioned that like this type of antibody would probably be administered through like IV infusions in humans.
Yeah, I believe most antibodies are administered via IV infusion.
Right, right, and yeah, like so, like how would the mechanism of a microglia antibody, broadly speaking, be different than the current FDA approved antibody Aduhelm? Because that one is also designed to stimulate the immune system to flush out these amyloid plaques in our brain, right? So can you give us an idea of like how they would work differently?
Sure. So an anti-A beta antibody like Aduhelm, the way that is designed to work is to target the plaque in the brain, to bind that antibody to the plaque, and then the antibody has a piece on it called a effector function, which is going to attract the microglia to the plaque, and they're going to chew up the plaque and eat it away. And the way that a TREM2 antibody would work would be a little different in that you would be targeting TREM2 on the plaque, or excuse me, on the microglia in like more of a way to turn on a signal. So it's not entirely clear how TREM2 gets turned on, like at a structural level. But what we think is going on is that if you get enough of the proteins in one area together, they sort of turn on that way. So if you sort of cluster them together, and one way you could do that is with an antibody, right? You could antibody has two arms, so you bring a bunch of TREM2s together and it turns on the signaling. And so in that way, you'd be sort of modulating how those microglia are responding to plaques and trying to make them respond in a favorable protective way to prevent, say, damage to surrounding axons or things like that.
Mm-hmm. Right. And I think maybe one of my last questions then is, the broader question here about, the amyloid hypothesis. And, there's a lot of questions now about, especially with the approval of Aduhelm, about like the evidence behind how effective it is to design treatments that with the ultimate goal of clearing amyloid plaques in people's brain, right? Like, does that actually help patients or not, right? If you do in fact design a drug that can clear out these plaques, but like what are the clinical effects for patients, right? I was wondering, what are your thoughts about that, and how can we like what do you think is how can we find out more about like whether that can really help patients, right?
Right. I think, with any disease, Alzheimer's is probably not that much different, where the earlier on in the disease you can target the disease process, the better off you're going to be. So, if you think of Alzheimer's and how the disease develops, if you start beginning to develop amyloid plaques, say 10, 15, 20 years before you would begin to exhibit cognitive problems. The earlier on you could target that process, the better off you'll be, right? And so with an amyloid drug, early detection using genetic risk factors or early biomarkers of the disease, which are being investigated, would be key to boosting success for preventing cognitive decline. And then as you would move further down that sort of disease process to where you start getting more tau pathology throughout the cortex and everything, it would probably become less and less effective. So, I think it's sort of the same with about every other chronic disease that can occur in people, is that the earlier you can intervene, the better off you'll be.
Right. And do you think that'd be the same case for like microglia-based treatments? The earlier is probably.
Yeah, I think, it's sort of the same thing. The less you have to push back against a disease process, the better off you're going to be, especially pushing back against an ongoing degenerating process. But again, it depends, how you would want to manipulate microglia might vary depending on where you are in that disease. You might want to do different things at different stages.
Right. Like if you want to, if you're designing a tau, like a microglia treatment to target tau, maybe that might have to be a little bit later, right?
Right. So if you wanted to, if again we take our preclinical data in the mouse, it would sort of suggest you'd want to tamp down that microglial response, or in the areas where you have this robust tau pathology. And that would be very different than wanting to sort of augment it in the case of amyloid. So.
Right. Got it. Yeah, that's, is there anything else you'd like to add about this topic?
No, I think it's an exciting time, and I hope good news keeps coming out.
All right. Well, thank you so much, Jason, for your time.
All right. Thank you very much.
All right. Bye.
Bye.